Come on. Recording. Okay, so we are today, Thursday, fall 2020, Bio 208 lab. We are going to be going over our heart material. And so the live stream here is to get through in the lab manual the major terms associated with the external, internal, histological features of the heart. And then after we get a little bit comfortable with doing it on the model and on the pictures, then we want to do the dissection. Chances are we're going to miss the dissection in the live stream just because that's going to be done individually based, and it's okay because your technique is not graded in a dissection lab and you do not have to dissect. Again, everything will be by picture or model come the lab exam. All right, so starting with, this is the heart, all right, and we want to get our surface features. Now, for our surface features, the heart is suspended in the thoracic cavity because of the great vessels that are on the superior top of the heart. So it's suspended down and it is kind of free floating in that pericardial cavity and then bigger the thoracic chamber. So when we think about what is connecting the heart to the rest of the body, it's the base, and the base happens to be the top of the heart where all the vessels are connected. So the base is going to be up here. The apex is going to be the pointy end towards the bottom. If we flip it upside down, the lungs, the base will be at the bottom and the apex or the pointy part will kind of go towards the clavicles up towards the top. So that's one of the differences in most of the organs. When we think of a base, we think of the bottom. In this case, it's the superior area where those great vessels are located. Okay. All right. Anterior part of the heart, you're going to normally look for the left atrium, the left auricle flapping over. You're going to look for that groove going down towards the point, all right? And the right auricle will kind of flip over, and the right ventricle will probably be more on towards the lateral side, all right? You're going to want to see the pulmonary trunk up front, the aorta coming over that pulmonary trunk and right pulmonary vessel, okay? And you're going to see these grooves. So when your atrias are inflated and full of blood, they're nice open chambers. But when there's no fluid in there, like a balloon without any air, they collapse and they kind of fold over the top. So that is going to create these kind of little grooves or ditches or canals that some of our vessels are going to come off of the aorta and run through. And those are going to be our coronary sulcus. Remember sulcus from the nervous system. Right? And again, where the cells are kind of coming into and curving in for the right and the left ventricle, we create another sulcus on the anterior part, and that is going to be known as our anterior interventricular sulcus because it's between the two ventricles. All right. And normally, again, in my experience in rat hearts, the right ventricle gets pushed off. So sometimes it's on the anterior and it's kind of almost midline and it looks like it's pointing straight down. Okay. Now, when we turn the heart over, and again, most of our models, you're going to see that the left atrium kind of has the big multiple pulmonary veins coming into it. All right. And you're going to see the enlargement of this blue coronary vessel, which is collecting blood to push it back into the right atrium. You'll see that blue vessel gets really close to this hole that represents our inferior vena cava. All right. And then there'll be a line, or again, a place where the invagination of the right and the left ventricle are taking place. And that is your posterior interventricular sulcus. Okay. So the importance of those sulci are that when blood is coming off of the aorta, and you can kind of see that a little bit, there's a little hole when you pull off, that's going to be your left coronary artery. And then there's a little right one over here. All right. The right one's here, the left one's here. They are going to come out to the edge. The left is going to immediately fork, and you're going to end up with the anatomically named anterior interventricular coronary artery, because where is it going? In the anterior interventricular coronary sulcus. And then the fork that goes this way in the left side coronary sulcus will be our circumflex, because it's going to twirl around. All right. The right one, again, it comes off right there and then connects right in here, it's going to roll around that right side coronary sulcus. And then there'll be vessels, another marginal artery that can come off. But those are the big arteries. And coming off of them are going to be smaller, smaller, smaller arteries that eventually are going to lead to the capillaries that are feeding the cells in the myocardium and then even the endocardium. Okay, and so that's how blood supplying oxygen, supplying fat, supplying proteins, supplying amino acids. That's how the cells here get those nutrients, because they can't get it from the blood in the chamber. All right, because remember that endothelial cell layer, simple squamous epithelium, is kind of preventing the blood from interacting with the underlying tissue, and we don't want the oxygen to start being exchanged in the chambers. Okay, all right, so that kind of gives us our sulcus. Again, our major branches of vessels. This front piece, when it's one, is known as the pulmonary trunk. It'll fork. One goes right, one goes left. Those are your pulmonary arteries, okay? The big red one up here, that's the aorta, and it'll get named for its segments. So it's ascending, then it's an arch, then it starts to descend, okay? The superior vena cava, I did that on Tuesday too. And then the inferior vena cava is this one, okay? So your major vessels coming off of your aortic arch, the first one is going for a little piece of time be one vessel that's going to eventually lead to some blood going to the right brain, some blood going to the right arm. So for that segment, it's known as a brachiocephalic artery because it's feeding head and arm. The second vessel coming off of the arch, the red one, is going to be going on the left side. That's your left common carotid. Okay? And then the next one coming off is going to be your left subclavian artery that's going to feed into the left arm. Okay? Now, the importance of the right common carotid and the right arm are linked together for a little bit. So if I ever want to try to stick a probe into my left ventricle, the best way to do that is actually to come through the left carotid. And if you look at kind of the geometry, I can kind of get into the left carotid, into that second part, and it's almost a straight shot into the aortic arch, down to the descending aorta, into the left ventricle. So when I used to do a lot of rat work, and we wanted to put a probe in the left ventricle to get pressure changes in the left ventricle, we would go in through the left common carotid. And those are small vessels. So it was tiny little probes going into tiny little vessels. And you didn't want to cut too much because then you cut the whole vessel. All right? Okay, so that's some of our major veins, major arteries, base apex. Okay? All right, so again, for your coronary arteries, all right? So the coronary arteries are going to be part of your systemic circuit. They are going to bring oxygenated blood to the cells and tissues. They are going to be collected by veins, systemic veins, and brought back to the right atria, okay? So again, coming off of the aorta, and in your picture in your lab manual, imagine if the pulmonary trunk is pulled off. The one little tubey red vessel here, that's the left common carotid. Not common carotid, that's the left coronary artery. Here's the right coronary artery on this side, okay? And again, your left is going to immediately when it hits the anterior part of the heart, at this little junction where the left auricle and the beginning of the anterior interventricular sulcus is, it's gonna fork. And one part goes down, one part goes around. So that's gonna be your anterior interventricular coronary artery. Sometimes in the literature about research, it's known as the left descending artery, and somehow left descending artery becomes abbreviated, the LED or the LAD. Don't ask me how, all right? But it's also sometimes nicknamed the widowmaker, all right? And so when people talk about having stents put in and they have a big occlusion that's preventing their left ventricle and the cells of the left ventricle from pumping effectively and pushing blood to their systemic system, this guy, this red vessel, the anterior interventricular coronary sulcus, the left anterior descending artery, that is your widowmaker, All right, and that's the one that if the occlusion is high enough and it's enough cells underneath that are dead and not getting oxygen, that your entire heart will basically infarct and you'll die. All right. All right. The right coronary is going to wrap around. It eventually will dive into the back and give us our posterior coronary, or posterior interventricular coronary artery. Okay. And then you should have, again, a red vessel coming off on kind of the sides when you're looking at anterior, posterior, and those are usually your left and right marginal arteries. Okay. Your big feeder for the left ventricle is this guy right here, and then the circumflex. And they're feeding, again, think they have their little capillaries pushing in to eventually feed all of these cells towards the inside, the papillary muscles, and the simple squamous epithelium that's lining that left ventricle. Okay. All right. Blue is going to represent usually deoxygenated blood. So the coronary vessels will be painted blue. They get names like great, middle, and small, right? Great, middle, and small. So the great cardiac vein is going to be the vein that usually runs with the widowmaker in the anterior interventricular coronary sulcus. All right. The middle cardiac vein is usually going to be the one on the posterior. All right. And your small is your other major cardiac vein, and the small, the middle, and they usually don't have the small in the models, but they're all going to merge into this big kind of sulcus. So enlargement that will kind of fill with the blood returning from the heart that's waiting to go into the right atria. And if you open up, and I don't know if you can see very well, but there's a blue circle in your right atrias that usually represents the opening of where the coronary sulcus, collecting all the blood from those major coronary veins, is going to release blood back into the right atrium with the inferior and superior vena cava. Okay? And that's how the blood going through the coronary circuit gets back into the heart to then go get reoxygenated with the rest of the deoxygenated blood heading into the lungs. Okay? All right, so that's your coronary vessels. There is another video on my YouTube page that goes in through the coronary vessels as well. Okay, the next thing we're going to talk about is histology of the heart. And with histology comes that review of our chapter 4 material, the specific tissues that are part of histological layers. So the heart's going to have 3 histological layers. It's going to have the innermost layer that is going to be epithelial tissue, those endothelial cells, those simple squamous cells that are running, again, throughout the entire circulatory system that are providing that barrier of blood pushing against them. And every epithelial tissue needs a little underlying connective tissue, that basement membrane, that lamina. So your endocardium histological layer is going to be a layer of endothelial cells and some underlying areolar connective tissue, right? And we expect some of the capillaries in that areolar connective tissue to be coming from eventually these coronary vessels, bringing oxygen, fat, amino acids, glucose to the cells so they can produce ATP and dump their carbon dioxide as needed. Okay. The next layer is going to be the big layer of muscle. All right. So it's going to be your myocardiocytes, your cardiomyocytes, all right? And remember the way these cells are, they are going to have striations. So they are going to be overlapping actin and myosin. And because they are just like skeletal muscle in that striations, we have sarcomeres. And we have the same proteins we expect to find in the sarcomere. So we're going to have troponin, we're going to have tropomyosin, we're going to have nebulin, we're going to have titin, and we're going to have actin and myosin. And actin is normally covered by tropomyosin, and troponin is attached to it. And actin and myosin can't interact as long as troponin and tropomyosin are covering actin's binding site. And the way to get troponin and tropomyosin out of the way is for calcium to bind. So that ties into why we talked a lot about calcium in our lecture the other day. Okay? Now, remember, these cells are single nuclei. They are small entities, and they will have like pieces and parts of breaks, and they'll fork into kind of like weird looking bow-like structures. So the connective tissue can provide room for capillaries to come between them. And then some of the areas where they're touching the cell next to them, they are going to merge their plasma membranes, produce special proteins that'll again make a channel that allows the cytoplasm in one cell to connect to the cytoplasm in another cell. So they are physically linked and chemically linked to some of their neighboring cells. They are areas touching other cells, but there will be some connective tissue that's going to force its way and make little spots between the cells so capillaries can be there. And where we have connective tissue, remember, we can also have collagen, elastin, fibers. And in the heart, you want a little bit of both. You want collagen because these cells, when they contract, we want them to pull on something so they can collapse the chamber. And at the same time, we want it to have a little bit of elastin because we want to be able to do this for 100 years, 80 times a minute, okay? And where do collagen and elastin fibers come from? Fibrocytes, fibroblasts. So in between the muscle cells, there, that little bit of space, there's going to have to be some fibroblasts, fibrocytes to create these collagen and elastin proteins. There's also, wherever there's connective tissue, the potential for macrophages, lymphocytes, mast cells, eosinophils, basophils, you know, all those immune cells. And there's also a potential for adipocytes, okay? And we don't want a lot of adipocytes in our connective tissues, especially in the heart, because do adipocytes, when they get filled with fat, do they have a lot of give and go? No. Okay, so be aware that the connective tissue behind the epithelium can have all of that, all right? And the connective tissue between the muscle cells in the little spots where it kind of gathers can have all of these different cells, right? And all of that connective tissue, if you think of it as a netting, like chicken wire, all right, that's what we build our cardiomyocytes around. And then when the cardiomyocytes contract, the chicken wire or the meshwork can collapse into a smaller size chamber and then can be pulled back open, all right? But we need something, because they're not attached to bone, so we need something to kind of have that ability for the force to be applied to, okay? All right, so that's your myocardial layer. And you'll notice in the left ventricle, lots more myocardium, okay? Than there is in the right ventricle. And then if you look in the atrial areas, there can be little to no myocardium. And that goes back to, well, a lot of how the fluid moves is by pressure gradients, and gravity is also helping. So unless you have a sticky valve, it would not be expected to have a very large muscular atrial layer, okay? But if you start having an AV valve that won't let fluid come through and that atria has to contract to push fluid in, it'll start to hypertrophy, okay? All right, now, going back to chapter 1 from last semester, you learned about serous membranes, and you learned that there is a thing called the visceral pericardial membrane, and then there's a thing called the parietal pericardial membrane, and there's a small gap between them known as the pericardial cavity. Okay, now you're going to learn that this outer, and in some of them it looks like a little bit of a different coloring, that is going to represent your visceral pericardial membrane, your histological layer known as your epicardium. Okay, and what are the tissues that make it up? Well, the outermost is going to be simple squamous epithelium, sometimes also known as mesothelium, that's producing a little bit of that fluid that's becoming part of the pericardial fluid. Under it, just like in the endothelium, but on the inside, it's a mix of areolar connective tissue, right, that lets capillaries, again, coming off all these coronary vessels, push in and provide oxygen, fat, protein, and release of, you know, the waste products there, okay? So the outer part of the heart is actually your visceral pericardial membrane, And with the membrane and that connective tissue, that is your epicardial histological layer. Okay. Now in our slides, we go into the next piece of tissue or lack of tissue is our pericardial cavity. Then you have to have the pericardial sac or the parietal pericardial membrane. And as it shows in the slides in your lab manual, there will be, again, another simple squamous epithelium. Then there'll be a areolar connective tissue. And then it'll get a little bit more densely collagenized to make a little bit more of an oomph to that sac. So that way, again, what is that outside sac potentially coming into contact with? Your bones, your periosteum, your perichondrium around your ribs, around the cartilage of your ribs, the lungs, and this parietal pleural membrane. And so you want to have, again, a little bit of that tough capsule to kind of help separate and protect the heart. Okay. The pericardial fluid comes from those two mesothelial or simple squamous layers. And remember, it's supposed to be a small amount. Every now and then on TV or in movies, they show blindly someone like jabbing a needle in someone's chest and sucking fluid out. And then all of a sudden the person's like, oh, okay, my heart's not like going crazy anymore. And they took too much fluid out of the pericardial sac and saved them. Well, if you miss that cavity, which is like that big, right? And you go a little bit deeper, where could you potentially shoot that needle in? The ventricle chambers. And if you're sucking that blood out, are you going to expect the person to get better? No, because now there's no blood pumping. All right. So whatever they show on TV of too much fluid in the pericardial sac, which again, like a water balloon, if it's too much fluid, it's hard for the heart to then push against and contract against. Too much fluid is bad, but you would never blindly go in and jab to pull that fluid out. That's just Hollywood for you. Okay. So review those histological layers. So if I ask you a question, what is the outermost histological layer? And I put the sticky right here, you would then say the epicardium. If I put it over on this red area and I say, what is this histological layer? That's the myocardium. If I put it inside somewhere and I say, what is this histological layer? That's the endocardium. If I ask you, I put the sticky right here and I say, name one of the tissues, specific tissues, part of the indicated layer. I'm not asking you to tell me it's the endocardium. I'm asking you for one of the specific tissues that is part of the endocardium. So then I expect you to tell me simple squamous epithelium, endothelium, or underlying areolar connective tissue. If it's here and I say, name one of the specific tissues found in the indicated layer, I want you to tell me that's cardiomyocytes, so cardiac muscle cells, and a mix of connective tissues providing a little bit of that cardiac skeleton to work against, right? And out here, again, you can say a specific tissue, underlying areolar connective tissue, or that mesothelium, that simple squamous epithelium, okay? So those are your histological layers, all right? For cells, again, review, what do cardiomyocytes look like? Again, they have those striations. They are single-celled. They are interconnected. The connections between the cells where there's more proteins binding the two cells to each other, letting them share cytoplasm. So they're sharing spit, okay? That is your intercalated discs, okay? And again, you can kind of see the fork in the picture that lets a little bit of connective tissue. That's where you would expect to see those collagen, elastin fibers, those capillaries, those connections that eventually lead back to your coronary arteries and your coronary veins. All right. Uh-oh, has Lauren been waiting? Sorry. Okay. All right, so now let's go into the internal structures and pieces and parts of the heart. Okay, so if I cut the heart on a sagittal plane, I make something right and left. So my right heart, left heart. If I cut on the transverse plane, I separate the superior inferior and I get two atrial chambers, two ventricle chambers. Okay, now the atrial chambers, when they're not full of blood, the tissue will just kind of deflate and it'll look kind of like flappy on top of flappy skin. So sometimes the skin that's flapping and hanging out and over when the atrials are not inflated, full of blood, will be called the right auricle and the left auricle. Okay, but remember, when you go to a room, an open space is an atria or an atrium. The flappy material, when it's not full of blood and open, is the right or left auricle. Sometimes they just get used interchangeably, because again, sometimes we get lazy as physiologists and anatomists. Okay, all right. You can't see it all that well, but where, again, the right coronary sulcus is letting blood into, there's a physical definition between what is the right atrium wall and what is the left atrium wall. And so you do have an interventricular, interatrial septum, okay? And sometimes you hear about kids, the septums don't fully separate. And so blood between the right and left atriums is flowing too easily, okay? Now, that's just the whole thing, okay? Now, keeping that there's a hole between the right and left atriums. In some of your hearts, you're gonna see a white kind of feature, and others see a little bit lighter white and short, okay? It's sitting in that interatrial septum. There is a hole that goes into this there when a baby is in the fetus of the mom, because the baby gets blood from mom that's oxygen rich, and blood from mom comes through baby's veins to the right heart. Baby doesn't understand all that oxygen rich blood and high amounts of the lungs are sitting in fluid. They're not functional inside the womb. So one way the blood can jump over to the left side quickly is through a hole between the right atrium and left atrium. And holes in bones are called framens, and it's like oval. So when you have a hole in the fetus, it's under formical valve. When you take your first breath as a born child, the pressure gradient is supposed to take and fold on top of that hole. So you close it, and you'll see a little bit more fully closed and seal. But that process of where it becomes a remnant of that hole, and that's connective tissue flat holding over it, that connective tissue we now call the fossa ovus. So in these adult hearts, that white feature, and they can do a little bit like much that is the remnant of the hole in the fetus, which is the holes there, the formical valve. Now it's the fossa ovus. Okay? Right. In the ventricular septum is going to be this big piece that separates the right and left ventricles. In our monoheart, the ventricle is pretty big, but in the sheep hearts, it's pretty thick. Our big ventricle is so pushed off to the side, right, that you don't usually see much of the chamber, much of the right side. Right? And this side is going to make a nice, almost skin-on figure. Right? Your valves separate the atrium ventricles from your atrial ventricular valves. They are the monoline valve, the tricuspid valve, monoline tricuspid valve. Again, the literature likes about some of the like so much like the literature is working on the valve, that valve, you refer to the valve, valve, your microscope has system, we try to push the valve. Right? If you the motif of the interior shell of a oyster, it's not smooth. There are cells there. Remember what we call them the column, of course, column measure to it, culture. No. So same thing we said about the ventricles, the column of the myocardial muscles kind of flipping around and forming a cave-like chamber. That's called okay? The right ventricle, because it's thin, you need to make sure that the evaporating components next to the coronary neck, where the crust of the valve gets to pull and keep more close, the ventricles will back into the right atrium. It needs to have connection to the wall on the outside of the right ventricle. So it's not very clearly that one distinct kind of band of muscle connects the atrium to the right side, right? And in other parts of the like the muscle system are and so you may make that electrically get electric signals contract all these outside muscles because they're so fluid compared to the left side. They don't want to make just the handling to the muscle in the ventricles, right? That's the right side, right? And the right atrial area sometimes muscle in the presented the muscle rolling like areas of out, so called actin muscle, right? Once arteriosus and goes to the valve. The follow one from the blood towards that valve. Now is that opening goes into the right ventricle on the other side, the valve, and the ventricle is clear in the valve. It's saturating the coronary and right ventricle. Right? If you want to have a little bit of funneling, so the conus arterius is going to be back here, right? Everything on there. So we normally talk about the senior valves, I'll just clear the right valve. We don't need to say coronary senior valve, right? And we don't normally put in the like there are valve and say that atrial ventricle. We talk about the hairs, we call them atrial valves, senior valves, because it's a another valve or you coronary senior valves. The difference between the atrial valves have to equal those. So that's why I have the coronary and the coronary is shown where valves. You see the columns that go in the caps that usually connect to your coronary and your muscles. Your valves are closing. They're pulling the strings to keep it closed, right? And then the over the close and the chamber is smaller. Hopefully the pressure of the fluid is smaller. Compartmentalized chamber, which is bigger than the other side of the valve, and you'll feel this happening, right? So that's basically all that has is in the lab manual for your external features. Again, it was a separate video on why you change from 4 coronary arteries. We're going to step down and roll the sections, tiny features. Sections are going to be more challenging to everything is collapsed in. Things is not as soluble. And it's almost embedded into the tissues, so it's really layer. You'll see much of the model. So a lot of times it's like, oh, well, let's guess what this is. Let's see where the probe goes and the probe goes to see where am I in the system, right? So use this again to review your lab studies, review your pictures, manual, and previous videos. Okay? So any questions before I